Hello, everyone, and welcome to today's live broadcast, Risk, Mitigation, and Management to Ensure Virus Safety of Biopharmaceuticals, Regulatory Perspectives and Industry Best Practices, presented by Dr. Hazel Aranha. We're excited to bring you this educational web seminar presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Sartorius. The Sartorius Bioprocess Solutions Division is a leading international provider of integrated solutions for the biopharmaceutical industry. With their extensive portfolio of products, technologies, and services, they cover wide areas of the biopharmaceutical process chain, ranging from fermentation, cell cultivation, filtration and purification, to media storage and transportation. For more information, please go to www.sartorius.com. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window, or use the Q&A box to let us know that you're having a problem. Now I'd like to introduce Dr. Hazel Aranha. Dr. Aranha is a biopharmaceutical industry professional with over 30 years experience in the industry, academia, and consulting. She is a well-acknowledged expert in the area of virus and prion clearance from biopharmaceuticals. She has provided consulting services in the U.S., Europe, and Asia. She teaches courses on ensuring virus safety of biologicals, as well as other topics including good clinical practices, GCP, good manufacturing practices, GMP, downstream processing, navigating the drug development cycle, and effective biomedical writing. Hazel has a master's degree in virology, PhD in environmental microbiology, and holds regulatory affairs certification, RAC, for both the U.S. and European Union. She has to her credit 2 books, over 45 publications, and 4 book chapters. Her past assignments have included positions at Catalent Pharma Solutions, Wyeth Vaccines, Pfizer, and Pall Corporation. Please join me in welcoming Dr. Hazel Aranha. I will now turn the presentation over to her. Dr. Aranha?
Thank you so much for that introduction, and thank you all for calling in to today's webinar entitled Risk Mitigation and Management to Ensure Virus Safety of Biopharmaceuticals. We will discuss today both regulatory perspectives as well as industry best practices. This slide is essentially an overview of our agenda for today's webinar. We will talk about whether virus safety of biopharmaceuticals is an attainable goal, then go on to regulatory approaches to virus safety, discuss some of the virus clearance technologies that are currently being used, talk about upstream risk mitigation approaches, and then close with a few concluding comments. Is safety of biopharmaceuticals an attainable goal, or is it nebulous? What are the issues that we have to address when we address the safety of biopharmaceuticals? To start with, why ensure virus safety of biopharmaceuticals? Short answer is patient safety. To ensure patient safety, we have to ensure that the virus, that there is absence of virus. Keep in mind that absence of evidence of virus is not evidence of absence. It is not possible to test for all viruses. Some mammalian-derived products, for example, plasma-derived products, do in fact pose a risk of virus transmission. For example, the HIV and the hepatitis viruses. Keep in mind that our blood safety profile has been enhanced considerably since the last 2.5 decades. Some cell lines, for example, the Chinese hamster ovary cell line or the CHO cell lines, do contain non-infectious endogenous retroviruses. Note, however, these are non-infectious and therefore pose only a theoretical risk of virus transmission. Adventitious virus may also be introduced through process additives and through GMP breach. Additionally, new and emerging viruses can enter our process stream primarily because of globalization. When we talk about globalization, it does not refer only to travel, but also to issues related to supply chain and raw materials, as well as trade and commerce. The next slide talks about the types of biopharmaceuticals that require virus safety assessment. Essentially, any product that has been directly sourced from human or animal-derived systems must be tested, and the virus safety must be documented. It is intuitively obvious that some materials, as for example plasma-derived products or coagulation factors which are directly sourced from humans, must be demonstrated to be safe. Other types of materials that are directly sourced from human or animal-derived systems include enzymes, enzyme concentrates, growth factors. Also, the recombinant products that are produced in continuous cell lines must be documented to have appropriate virus safety. In addition to those products that are directly sourced from human and animal systems, any product that has had indirect exposure to human animal-derived components must also be assessed for virus safety. This includes, for example, supplementation of cell lines with animal-derived growth factors, reagents used, for example, in cell line establishment, as well as even antibodies that are used during clonal selection. More recently, we also see novel production systems, as for example, avian cell lines. The next slide essentially covers the kinds of sources of viruses during biopharmaceutical manufacturing. For example, bovine serum and bovine serum albumin used to be extensively used during biopharmaceutical manufacture. This poses a risk of viruses such as bovine virus, diarrhea virus, BVDV. Porcine-sourced trypsin was in fact used in cell line establishment. Other areas, for example, that are required to be addressed include Chinese hamster ovary cells, and as we mentioned earlier, these are endogenous retroviruses. However, they are non-infectious, and we therefore need to incorporate adequate virus safety assessment when working with these cell lines. Additionally, viruses can also enter the manufacturing stream adventitiously, as for example, mouse minute virus, which has been a documented contaminant in bioreactors, and also they could potentially be cGMP breach during manufacturing. The next slide is essentially an overview of some of the virus and prion contamination incidents. Viruses have been associated with plasma-derived products, and also in the past we have, for example, had concerns with vaccines being contaminated with, for example, poliovirus, rabies virus, rabies vaccine, and yellow fever vaccine. Note that in all these cases, virus contamination was not due to the negligence on the part of the manufacturer, but because detection methods were not sensitive enough to detect the viral contaminant. The next slide essentially addresses the question, are recombinant products safe? Short answer, yes. There has been no reported transmission of pathogenic infectious virus to humans from a recombinant product. That said, contamination can and does occur. The production systems could be potentially contaminated. One example here is the rotavirus vaccine that was shown to be contaminated by porcine circovirus, or PCV. This virus was traced back to the porcine trypsin that was used in cell line establishment. Once again, I'd like to emphasize that the contamination with PCV was not due to the negligence of the manufacturer, but because our virus detection methods were not sensitive enough at that time. Additionally, there could be cases of adventitious virus contamination, either of the bioreactor itself or alternatively facility contamination incidents. What are the consequences of a contamination event? Key is the fact that it has the potential to impact patient safety. These drugs are a necessity for patients, so drug shortages do in fact impact the patient when there is a contamination event. Other issues relate to regulatory and legal implications, as well as financial losses and lost opportunity cost. In the next section, we go on to regulatory approach to virus safety and risk management. The general approach to virus safety of biologics is a multi-pronged approach that includes appropriate sourcing and testing of raw materials and cell lines, an adequate risk assessment, validation of multiple orthogonal virus clearance technologies, and in-process testing. Keep in mind that it is impossible to claim absolute absence of virus presence. The best we can do is use a pragmatic approach to virus safety assessment. This slide demonstrates the sources of viruses, potential sources of viruses. For example, it's important to control the starting materials. Plasma-derived products are extensively tested. In addition, there is donor selection, exclusion, and so on. Keep in mind that, once again, no plasma pool that is tested positive for any virus would be included during the manufacture of plasma-derived products. However, once again, if there are low levels of pathogenic virus, they may escape detection. During the manufacture of recombinant products and monoclonal antibodies, there's an extensive multi-tiered approach that involves cell bank establishment, in other words, establishing the master cell bank and the working cell bank. These cell banks are extensively tested and are an absolute requirement from a regulatory standpoint. In addition, it is important to control the manufacturing process through control of additives. For example, serum was a common supplement in the past. However, the industry has moved away from supplementation with serum. Please keep in mind that serum-free does not necessarily mean animal product derived free. Additionally, purification reagents such as monoclonal antibodies as well as excipients could also potentially be a source of virus contamination. The next slide talks about raw materials related challenges, and there are multiple challenges when sourcing raw materials. To start with, any biopharmaceutical operation involves a large number of components. These include both chemically defined and complex materials derived from either animals, plant, or bacterial sources. Additionally, when sourcing raw materials, there are a large number of suppliers, which again requires considerable vendor oversight. Traceability of raw materials is a concern, and as we mentioned earlier, currently the focus is on global sourcing. Some countries have a very stringent quality system in place, other countries less so. There may also be variability in raw materials, even with stringent quality management practices in place. As we said, there are testing-related issues. Contaminants are not homogeneously distributed. Therefore, detection is based on probability, and low levels of virus may go undetected. In terms of materials impact, even though materials do in fact meet specification, they may, some materials may impact the final drug product quality. The next slide essentially addresses our concerns with virus detection methods. Essentially, you only find what you are looking for. For example, even though PCR is a very sensitive method, it only detects specific virus primers. Also with infectivity assays, once again, specific cell lines are needed to detect viruses, and this may pose a concern. Additionally, sensitivity of assays is a concern in terms of the limit of detection. Low levels may go undetected, especially when you have large volumes of raw material or products. Also, even when testing is conducted, there are sample volume limitations. Other issues to consider when talking about virus detection methods include false positives and false negatives. The next slide provides you with an overview of risk evaluation in terms of sourcing and production considerations. The factors to keep in mind are the type of product, the production system, as well as raw materials. For example, in terms of the type of product, directly mammalian-sourced products such as plasma-derived products obviously pose a higher risk of contamination as compared with a recombinant product or a monoclonal antibody. The kind of production system used, once again, factors into the risk. Well-characterized cell lines, for example, the Chinese hamster ovary or CHO cell lines, pose a significantly lower risk as compared to, for example, advanced therapy materials, ATMPs, for example, gene therapy products. Transgenic systems, if appropriately controlled, pose a low to medium risk. With regard to raw materials, animal-derived additives, as we mentioned, pose a high risk, whereas indirectly animal-derived systems supplemented with, for example, beef or meat extract could pose a low to medium risk. In terms of the regulatory approach to virus safety, a holistic approach is recommended. For example, risk management is based on a triad approach that involves risk minimization or prevention through appropriate sourcing, incorporation of robust virus inactivation and removal steps, and this has to be documented through process validation, and testing during production to ensure absence of contaminating virus, or in other words, in-process testing. What does this mean from a practical standpoint? One question that's frequently asked is how much log clearance is needed for a regulatory submission. Unfortunately, there is no absolute value. Log reduction factor required is determined on a case-by-case basis. Factors for consideration are risk associated with the starting material. For example, a murine-derived cell line poses a lower risk as compared with a human cell line. Similarly, serum-free is a lower risk as compared to media containing serum. Other considerations are, for example, the product indication and patient population. A young child with a mild disease is a higher concern as opposed to a terminally ill patient. Dosage and frequency of administration is another concern. For example, large multiple doses are a higher concern as compared with one-time small doses. One example here is coagulation factors that may have to be administered with higher frequency as compared to a small one-time dose. This slide essentially provides you with the regulatory requirements both for marketing authorization applications as well as for investigational medicinal products. The regulatory references are at the bottom of the slide. The point I would like to make on this slide is essentially that with regard to the validation with an entire panel of viruses, the entire panel is required at phase 3 or when the marketing authorization is submitted. The entire panel is not needed in the case of investigational medicinal products. A more streamlined virus validation approach is acceptable. Why conduct virus clearance evaluation studies? The objectives are twofold. One is to evaluate the ability of the manufacturing process to clear, that is, in other words, inactivate or remove known virus contaminants. In this case, for example, we could think about viruses such as HIV or hepatitis viruses with plasma-derived products and retroviruses in the case of cell line-derived products. Additionally, the objective is also to estimate the robustness of the manufacturing process to clear not just known viruses, but unknown viruses as well. We apply over here a model virus concept, which is the use of relevant and specific model viruses, as well as non-specific model viruses. The next slide provides you with an overview of a validation study design. I could spend an entire webinar just discussing validation study design, but at this point, just to give you a brief overview with regard to this, the way the study has to be designed and conducted. Essentially, determine your clearance objectives based on risk assessment. For example, in the case of plasma-derived products, there is a higher risk potential, so there will be a higher virus clearance requirement. Identify critical parameters and set acceptance criteria. Depending on the particular unit operation, the critical parameters will vary, and we will talk a little more about it in some of the upcoming slides. Accurate process modeling is essential. The test virus panel included must be appropriate. For example, there is a relatively standard panel for plasma-derived products as well as for monoclonal antibodies and recombinant products. It is also essential to determine the clearance mechanism, whether it is inactivation or removal. This is a regulatory requirement. Some steps may provide both inactivation and removal. In this case, it is essential to document what part of the clearance was due to inactivation, perhaps due to the low pH buffer, or removal, perhaps due to the chromatography column. Ensure the product validated is representative of processed product in terms of composition, that is purity, potency, and yield. This would appear to be a no-brainer. On the other hand, you will be surprised at the fact that there are times when virus clearance studies are scheduled at contract testing labs, and for some reason, the product that has been shipped over to the contract lab may not necessarily be representative. It is important to note that studies are typically done with operating parameters set at worst-case conditions. So this gives you an example of the scaled-down models and the kinds of parameters to address for both when scaling down from production scale to bench scale. For example, if we consider virus removal filtration, some of the factors to consider are the protein concentration, which should be worst case or the highest protein concentration that you anticipate, the operating pressure, throughputs, flux, temperature, whether the testing, whether the manufacturing is being done at room temperature or in the cold. For the most part, most manufacturing processes are done at room temperature. In addition, pH as well as ionic strength has to be representative as well. The next slide talks about the categories of viruses that need to be included for virus validation studies. A relevant virus is a known or suspected virus or a virus of the same species as the contaminant virus. An example here in the case of plasma-derived products would be viruses such as HIV or hepatitis A virus, HAV. Specific model viruses are also included. These viruses are closely related to a known or suspected virus and have similar physical and or chemical properties. Additionally, to document the robustness of the manufacturing process, non-specific model viruses are included. The next slide gives you an example of the kinds of viruses that are included for phase 1 and phase 3 spiking studies. As I referred to in an earlier regulatory slide, if it's a phase 1 study, a small virus such as a parvovirus is included, as well as a large virus such as either xenotropic murine leukemia virus for recombinant products or HIV for plasma-derived products. Parvovirus is considered worst case because it's a small virus and it's non-enveloped, difficult to inactivate using inactivation methods, and also it's a relatively small virus. The next slide essentially gives you specific characteristics of these various viruses that are included in virus validation studies. The virus validation studies are essentially performed in 2 phases. You have to ensure that any inactivation or removal of virus clearance that you see is not associated with the matrix. We therefore perform cytotoxicity and virus or matrix interference studies to determine whether the product intermediate is toxic to the indicator cells. Alternatively, does the product intermediate contain substances that would interfere with virus detection, or does the matrix of the product interfere with the detection of viral nucleic acid by PCR? In terms of a virus clearance study, the test article is spiked with the appropriate virus and exposed to the unit operation, be it virus removal, filtration, or chromatography, or low pH. Fractions are then collected during the unit operation and assayed using either infectivity assays or PCR. This slide is essentially a schematic documenting the kinds of controls that you would need to include. For example, you include a control to determine that there is no virus inactivation in the sample matrix itself, and also controls to document that there is no virus inactivation over time. We will now get a bit specific and talk about the kinds of viruses that are virus clearance technologies that are currently used in the industry. Before discussing virus clearance methods, I thought it would be good to briefly look at the specific properties of viruses that are important to us from a virus clearance standpoint. Note that this classification is obviously an oversimplification, but in terms of virus clearance methods, we can classify viruses based on size, which are the large viruses, for example, herpes simplex virus and pseudorabies virus, as well as small viruses such as parvovirus B19, mouse minute virus, porcine parvovirus, and hepatitis A virus. Viruses can also be classified based on the presence or absence of a virus envelope. And examples of enveloped viruses are xenotropic murine leukemia virus, HIV, HBV, HCV, cytomegalovirus, and influenza virus. Keep in mind that when methods are used specifically targeting the enveloped virus, it impacts the surface envelope, and the virus may not be rendered non-infectious. However, the virus is not able to replicate. Examples of non-enveloped viruses are parvoviruses. These viruses in general are highly resistant to inactivation methods such as heat, acid treatment, and so on. In terms of our virus clearance toolbox, virus clearance methods can be divided into virus inactivation methods such as heat and irradiation, or virus removal methods such as filtration, chromatography, and precipitation that was extensively used in the plasma industry. Note that methods must be orthogonal. In other words, they must have different mechanisms of action. So multiple orthogonal mechanisms must be incorporated. The next slide is essentially an overview of steps used in downstream processing. Virus clearance unit operations may be part of routine downstream processing. For example, the protein A step as well as chromatography steps. Alternatively, they may be specifically incorporated into the manufacturing process to estimate the virus clearance capability of the downstream process. The virus inactivation or clearance is expressed as log reduction value. The next slide essentially provides an overview of the kinds of steps that are effective for both non-enveloped viruses as well as enveloped viruses. Note that steps such as detergent inactivation and low pH inactivation are only effective against enveloped viruses. A method such as virus removal filtration is considered a robust method because it acts based on size. In addition, it's effective against both non-enveloped as well as enveloped viruses. Some chromatography steps, for example, anion exchange chromatography, is considered more robust than some of the other steps, such as cation exchange chromatography and hydrophobic interaction chromatography. Note that these steps, such as chromatography and heat inactivation, can act both on non-enveloped as well as enveloped viruses. However, the robustness of the step depends on process conditions. This slide is an example of log reduction values obtained in virus clearance studies. So, for example, several of the steps that are routinely used in the industry and for which virus clearance could be claimed are steps such as protein A treatment, either a low pH or a detergent inactivation step, and anion exchange chromatography, affinity chromatography, and virus filtration. Keep in mind, for example, methods such as low pH inactivation as well as virus removal filtration that has a greater than or equal to sign in front of the numerical value indicates that no virus was detected in the effluent aliquots tested. Note also with low pH inactivation, since MMV is a non-enveloped virus, low pH inactivation was not determined on the using MMV as the test virus. The cumulative log reduction value for the entire process has to be calculated. In this particular case, greater than or equal to 19.58 logs was obtained for xenotropic murine leukemia virus, and greater than or equal to 10.24 logs was obtained with mouse minute virus. We will now get a little bit specific, and using virus filtration as one of the examples, go into some details with regard to key considerations when incorporating virus removal filtration. The current industry standard is to use a 20 nanometer nominal pore size filter because the removal is primarily by size exclusion. Virus removal filtration is targeted against a broad range of viruses. In other words, it does not matter whether the virus is enveloped or non-enveloped. Removal is based on size exclusion. It's non-destructive, unlike some methods such as heat inactivation, which could potentially impact the proteins, the therapeutic protein. And also virus removal filtration maintains the biological activity of the process. It's a robust and efficient virus clearance step, and it's scalable and validatable. Regulators often recommend that virus removal filtration be incorporated as one of the steps during manufacture of protein therapeutics. Filtration, where size exclusion-based removal is the predominant mechanism, is a reliable and robust method for virus clearance. This next slide essentially shows you the cutoff of a 20 nanometer nominal pore size filter and the sizes of some of both the therapeutic proteins as well as the viruses. The non-enveloped viruses are in the yellow and the enveloped viruses are in the green. Note that parvovirus is at the cutoff of the 20 nanometer virus filter, so it is essentially a challenge to ensure that removal of parvovirus is attained while at the same time obtaining high product recovery. This slide essentially goes into details about the Sartorius virus filter portfolio. The virus-out filters that can be used for monoclonal antibodies and recombinant products are essentially the virus-out HF or the virus-out CPV. The membrane is a PS membrane. Depending on the feed stream, either the virus-out HF or the virus-out CPV could be used. The virus-out HF is a high-flux, high-capacity filter, provides high performance, and is also has high pressure resistance. The virus-out CPV is once again provides, like the virus-out HF, high virus retention. Both provide more than 4 logs reduction. The virus-out HC is a double-layer PES, which is suitable for hydrophobic proteins and was developed primarily for the plasma products industry. Again, this provides more than 4 log reduction of robust virus clearance. The virus-out HC is also a low fouling membrane, so it does in fact provide high product recovery. The virus-out Max is a pre-filter that is designed specifically to protect the virus removal filter that's, for example, the HF, the CPV, or even the HC. The virus-out Max is a triple layer optimized polyamide filter, and anecdotal as well as field data documents that if the virus-out Max is used as a pre-filter to any of the virus removal filters that are incorporated downstream, more than 4 to 10 times throughput can in fact be obtained. The virus-out media filter is a risk mitigation filter, and we will talk a little more about the virus-out media filter when we discuss risk mitigation. When considering virus filtration, what are the issues that have to be addressed? The process solution characteristics must be evaluated to ensure that there is ideal throughput through the virus removal filter. In some cases, premature fouling or blockage could potentially occur. This could be either due to process impurities, such as protein aggregates, denatured protein, host cell protein, and so on present in the process stream, or alternatively, it could be a result of the high molecular weight or the hydrophobicity of the protein. Note that when considering virus removal filtration, there are more considerations than just flux and throughput. This next slide essentially shows you how incorporation of an adsorptive pre-filter such as the Virusart Max can in fact significantly improve the throughput and the capacity through the Virusart CPV filter. Factors that could potentially impact virus retention performance of virus removal filters. Keep in mind that we would like to think that virus removal by size exclusion is based only on size exclusion. However, this is not the only mechanism that comes into play during virus removal. Parameters known to cause small virus breakthrough are concerns such as flux decay and throughput. Issues such as process interruption and pressure release, as well as low and high pressure, have been shown to impact virus retention. Other concerns relate to protein concentration, buffer composition, as well as virus spike purity during the virus validation study. I'd like to emphasize on this slide that each brand of 20 nanometer virus retentive filter has a specific log reduction value versus flow decay and a throughput profile. Worst case conditions are different for different brands. The next slide shows you data related to pressure interruptions on the log reduction value of the Virusart HF and the Virusart CPV. 5 Virusart lab modules were tested in each case, and multiple fractions were collected. The first fraction was the first 15 milliliters. There was then a pressure pause for 5 minutes. Following that, the next fractions were collected at a 2-bar operating pressure. Note that consistently high virus retention was obtained, and any differences in the bar graphs are only a limitation due to the detection method. As indicated at the bottom of the slide, no virus was detected in all aliquots tested. All log reduction values are greater than the numerical value reported in the graph. The next slide shows the independence of high and low operating pressure on the log reduction value of Virusart HF and CPV. Once again, operating these filters at 0.1 bar, 0.3, 1 bar, and 5 bar had no impact on virus removal. Once again, I would like to emphasize that no virus was detected in the tested aliquots. All log reduction values are greater than the numerical values reported. We will now go on to the next section that talks about risk mitigation and upstream operations. This essentially is a schematic of virus reduction in bioprocessing. Downstream virus clearance is an absolute requirement. It's a regulatory requirement to ensure patient safety. Upstream risk mitigation is not a regulatory requirement. However, it does make business sense. This slide essentially summarizes some of the contamination incidents that have occurred in the industry. Note, however, that these contamination events occurred either in the bioreactor or the facility and did not impact patient safety. In terms of risk mitigation approaches, obviously the primary emphasis should be on quality, prevention through assessment, inspection, and control of incoming raw materials. In addition, process intervention approaches have also been used. For example, upstream barrier technologies like gamma radiation, high temperature short time, and filtration. This slide is a general overview with regard to the mitigation approaches used in the industry. I'd like to point out some methods such as high temperature short time and gamma radiation are physical methods that could potentially impact the quality of the cell culture media that goes into the bioreactor. More recently, filter manufacturers have developed virus removal filters for upstream virus removal. Note that the virus filtration membranes are applicable primarily to chemically defined media. This next slide gives you an overview of Sartorius' Virusart media filter, which is a PES membrane, provides high flux. It has no impact on cell growth or product expression. Note, however, this has to be determined by the user. In terms of retention, in addition to greater than 4 log reduction for small non-enveloped viruses, the Virusart media filter also provides greater than 7 log reduction for Mycoplasma, Brevandomonas diminuta, which is the indicator organism for sterile filtration, as well as Leptospira. Leptospira has come to our attention recently because of a contamination event in one of the biomanufacturing facilities. This slide provides you with throughput performance with some commercially available cell culture media. Several of these media are currently being used in the industry. With regard to virus removal studies, Sartorius has conducted testing with 2 different types of media, the Pro-CHO 5 as well as the SAFC Excel CD CHO 3. Note that consistently complete clearance of the virus was demonstrated. To wrap up this webinar, I'd just like to conclude with a few comments. As we said, it is impossible to demonstrate absolute absence of virus presence. Regulatory and industry groups recommend a pragmatic approach to virus safety that includes appropriate sourcing, validation of the downstream process, as well as in-process testing. That said, virus safety assurance will continue to be a moving target due to several challenges. For example, the increasing complexity of our starting materials and production systems. Known and unknown viruses do have the potential to compromise biopharmaceutical safety, and as we mentioned, there has been increased globalization, which then impacts introduction of new and emerging viruses into susceptible populations. That said, there has been no incident of virus transmission to patients through recombinant proteins or monoclonal antibodies. This is the result of continued vigilance through an integrated strategy that includes incorporated robust quality management systems, control of entry of adventitious agents through prevention and intervention approaches, incorporation of unit operations that provide robust virus clearance, and in-process testing and environmental monitoring programs. To sum up what we've just discussed in this webinar, this quote is very appropriate. The weakest link in the chain is also the strongest. It can break the chain. Thank you very much for your attention.
Thank you, Dr. Aranha, for that informative presentation. It is now time for Q&A. If you have a question you'd like to ask Dr. Aranha, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click the send button. We will try to get to all of your questions, but if not, we'll be sure to follow up with you after the broadcast. Let's get started. Our first question is, Dr. Aranha, do you always need a pre-filter upstream of the virus filter?
Short answer, no. While a pre-filter is not a requirement, it will improve your process economics. The choice of the pre-filter is important. Pre-filters can remove aggregates either through size exclusion or alternatively through adsorption. There are currently filters on the market, for example, the Virusart Max that I recently discussed in an earlier slide that removes aggregates by a combination of both the mechanisms.
Dr. Aranha, can I use a downstream virus clearance membrane for upstream filtration of media components?
Yes, absolutely. However, once again, during manufacturing, in addition to the performance of the filter, you have to keep in mind as well the process economics. So you can use the downstream membrane such as the Virusart CPV or the Virusart HF or any of the other filters on the market, but the cost in general is usually about 10 to 20 times higher, and your flux rates as well will be much lower.
Now, although prion are not exactly viruses, how does the pharma industry detect or clear prions?
Well, prions are a concern primarily for the plasma-derived product industry, whereas for the biotech industry, it is a theoretical concern. That said, even with prions, we use our multi-pronged approach in terms of risk assessment, risk mitigation, and management. For right now, we do not have any significant concerns, especially with biopharmaceutical products or recombinant products. And I would be glad to address this question in greater detail offline because I could perhaps do an entire webinar just on these issues.
Dr. Aranha, what is the accepted virus load for products for some popular products that you know of?
Would you repeat that question? I didn't quite get that.
Sure. We have an audience member who is asking, what is the accepted virus load for products for some popular products that you know of?
Oh, okay. Well, actually, once again, this goes back to one of the earlier slides which discusses the amount of log virus clearance that you need. The important aspect is to ensure that you determine the baseline virus load. In the case of plasma-derived products, it is intuitively obvious that they will not have any detectable is the key word, detectable virus load for a variety of reasons. With plasma-derived products, it is an actual concern. With recombinant products, the cell lines such as the CHO cell lines do in fact have endogenous retroviruses which are non-infectious. So the regulatory requirement is to determine the baseline retrovirus load. This is very often done by a method such as transmission electron microscopy, TEM. Once again, I emphasize that the baseline load is non-infectious, and the regulatory and industry expectation is that once that baseline load is determined, you need to incorporate 4 to 6 log safety factor into the process.
Okay, let's see. I believe that is all the time we have for today for our questions. If we did not get to your questions, we will be sure to follow up with you after the broadcast. Dr. Aranha, do you have any final comments for our audience?
Yes. Once again, just to emphasize, the industry applies a very pragmatic approach, as I mentioned, absence of evidence is not evidence of absence. Our safety assurance will be a moving target, so we have to be continuously vigilant and incorporate multiple orthogonal methods. If there are any other specific questions, you can send it either to me or to one of our local Sartorius representatives. I would be very glad to answer them. Thank you.
Thank you again, Dr. Aranha, and thank you for Sartorius for making this presentation possible. I want to thank everyone for joining us today. I want to remind you that today's webcast will be available for on-demand viewing through February 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. We hope to see you again soon. Goodbye.
Thank you.